Welcome to our channel where biology meets Clyde. This is part 2 of the Dbt-Bet series where I will discuss 5 repetitive and critical previous year questions on cell biology. Whether you are an expert or a beginner, stay connected with our series to gain a deep understanding. And if you haven't watched our part 1 video, then the link is in the description box. Go and watch it. And so, let's get started. An integral plasma membrane protein is glycosylated, and scientists can trace the journey of this protein from the time of its initial translation. So, when the initial translation will happen, so after the maturation of the mRNA, it will come out to the cytosol, that is the mRNA, and here it will meet with the ribosomal protein, and thereafter it will start producing the target protein. So that would be the initial translation to its translocation to the plasma membrane. So the translocation. So thereafter it will enter to the ER membrane, and thereafter it will go to the Golgi apparatus, and thereafter it will be packed and reach to its location, that is the plasma membrane in its mature form. So that is the initial case, and at the end it will be matured and entered into its location, that is the plasma membrane. Which of the following changes in its molecular weight can be expected to take place during the maturation? So here, when this matured mRNA is present in the cytosol, and after the incorporation with the ribosome, it is producing the target protein. So here, the N terminal of this protein, like if that is the protein, so the N terminal should be synthesized in the first case. In earlier, the protein should be synthesized from the N terminal part. That is the natural phenomenon. And here, in the N terminal, you will get a sequence that is called as the signal sequence. So it is having a specific kind of sequence, like so this will be, so this should stay at the N terminal end, and there is a protein called the signal recognition protein. So that should come and bind to it. So it is a ribonuclear protein. It has a sequence that is complementary to this N-terminal part, which is having the signals peptide. It will come and bind to it, and also it will have a linkage with the, like if that is the N-terminal part. So the uh, that signal recognition protein will come and bind like this. So it will also have a bonding between the N-terminal signal peptide and also with the ribosome. Now the translation will be blocked for that period, for some period of time, when cause the SRP, the signal recognition protein, is here. So now it will bring it here. So here is a SRP recognition protein. So it will come and bind here, and after the successful binding, this SRP protein will be, again, it will be hydrolyzed. The GTP hydrolysis will be there. So in the binding form, it is in the GTP, and when it will come and reach to the SRP recognition site, that is here, so thereafter the GTP hydrolysis will be there, and the SRP will transform into the GTP form, which will again go back to the cytosol, and thereafter it will continue a new cycle to bring another complex, like to bring another mRNA and protein and this ribosome complex to the translocation site to the ER membrane. And now there you can see this is the N-terminal signal sequence. So here is an enzyme that is called the signal peptidase that will cut the site here. Now you can see here that signal peptidase and the mature protein has been, like, there is a separation, you can see. So there is a signal peptidase which will cut the end of the signal peptide. So here thereafter the coming that coming protein will be matured. So in the initial case, the premature protein will have the signal sequence, but the signal peptide is well cut. And now, so this signal peptide is initially it is having a sequence of 16 to 30 amino acids are there. So if there is a cut, that means you are losing the molecular weight. You will lose a sufficiently significant weight because it is having the 16 to 30 amino acids long strength. So here, so after the cut down of here, so why this signal peptide is present in the that inside the that in the integral plasma membrane. So cause so in that 16 to 30 amino acids, 6 to 7 amino acids are hydrophobic in nature. That's why they are not getting on the cytosolic part. They are bounded inside the non-polar position. So now, so soon after the work of this signal peptidase, so here is a protein that is called the dolicle. This dolicle will come here and attach a that will glycosylate the protein at the asparagine residue. So here the attachment will be of that NAC to NAC 9 mannose and 3 glucose. So it will come and bind to the asparagine residue. This asparagine residue will form a n-linked glycosylation at this asparagine residue. So this kind of glycosylation is very much important to be recognized via the chaperone proteins. That is BIP is a example of a chaperone protein. So this protein will come, and if the glycosylation is there, so it can come and attach with this protein, and thereafter it is very much important to give the protein to a specific kind of folding so that it could be in a so that it can form a functional protein. So the chaperone is very much important to give it give it a specific kind of structure. So the chaperone, so to be recognized by the chaperone, this kind of glycosylation is too much important. So now, and thereafter it will go to the that is the Golgi complex, and here this is the cis position, and so here the O-linked glycosylation will happen. Again the glycosylation will be happening here, and that would be the O-linked. Here the N-linked glycosylation was here, and here it will be the O-linked. So that will go via this way, and thereafter it will be matured and it will be packed into the carboprotein and will go into the its specific location, that is the integral plasma membrane position. So here so far what we have been seen, so that is in the first case, so it will lose its that signal sequence. So in the initially it will decrease, the molecular weight will be decreased, and thereafter the glycosylation will happen. So first time the glycosylation will happen in the ear lumen, and thereafter it will again happen inside the Golgi vesicles. So thereafter it will increases. So the option 2 is correct. The list 1 with list 2, here you can see the name of certain proteins. So we will start from the first one, that is the SRP. SRP is known as the signal recognition protein. We already have seen this SRP protein in our first question. So here you can see the same story, that is the signal sequence you can see here, and the translation is happening in that image, and it will be anchored with the signal recognition protein. It will bring it to here, the signal recognition protein receptor, and thereafter the GTP hydrolysis will be there. So the SRP will be GTP in a SRP, this SRP in a GTP form. It will again go and bind to the next target. So thus the cycle will go on, and we already seen that, already have seen that this is a ribonucleoprotein. So the ribonucleocomplex to bind to the signal peptide is the signal recognition protein. The next one is the PTI, that is known as the protein disulfide isomerase. So this protein here, this protein via oxidation, it can form the disulfide bond, and via reduction it will remove the disulfide bond. So inside the ear lumen, this protein is present in a abundant amount, that is very much important, very much crucial to form the disulfide bond in the protein. And it also can form the isomerization. It's in the name, the protein disulfide isomerase. So via the isomerization, it can regulate the degree of the disulfide bonds that is present between the two cytosine residues where the free SH. The cytosine, the free SH is there, and they will form the disulfide bond in between. And here, so that PTI protein, the protein disulfide isomerase, it is also having a free, that you can see, to free SH group. Via this, they regulate the oxidation reduction or the isomerization to form the disulfide bond in the target protein. So it will go the formation of correct disulfide bonds in protein. That will be the PTI. And next it comes the BIP. BIP also we have seen in the first question. So this is the BIP protein. So BIP stands for binding immunoglobulin protein. That is a specific kind of protein that is present in abundant amount in the ear lumen. So that is a chaperone protein. It will come and fold the protein in certain way so that it can get its exact tertiary structure. So the chaperone in the ear lumen is the BIP. Now comes the anchor protein, that is GPI, that is known as the glycosyl phosphatidyl inositol, GPI. So what is the glycosyl part? This part is the glycosyl part. So here the 3 mannose, you will get 3 mannose and 1 glucosamine, and this is the phosphatidyl inositol, that is the phosphatidyl part. So that is a chain of phosphate group, and here you will get the inositol in the surface. So that is hydrophilic, and it is the hydrophobic part. So inositol you will get here, and it is having a specific structure, that is the glycosylated phosphatidyl inositol you will be getting. And now there is another thing, the ethanolamine. So that is the whole total structure. So here the phosphatidyl inositol, here the glycosyl part, and here the ethanolamine. So this one is having a free in each group, at the amino group on its terminal, and it makes a bind with the C terminal of the target protein, that is having a that carboxylic group, and they will form a amide bond. So thus by forming this amide bond, so that target protein will be anchored on the plasma membrane. So plasma membrane has a specific structure, this one, that is known as the GPI, and it will form a amide bond between its target and this thing itself. So thus the protein will be attached here. This will be anchored here. So here the term comes a lipid anchor of proteins attached with the membrane. So this will be the fourth one. So the option 3 is correct. So the following process turn on and turn off the alpha subunit of a heterotrimeric G protein. So this is the very common structure of the G protein, that is a 7 transmembrane protein, and it is associated with the heterotrimeric region. So heterotrimeric, three separate kind of proteins are there. One is alpha, beta and gamma. This when it is in the resting situation, that is the resting situation. When they are in a resting phase, so the alpha, beta and gamma, all the proteins are associated, and they are also associated with the that all these trimeric proteins are associated itself, and they are binding, they are bounded with the G protein. And now if a stimuli comes, like a ligand comes, here in the picture you can see a hormone. So if a ligand comes and it binds to the receptor, so the conformational change, the protein conformation will be changed in a certain way. So here in the resting phase, the GTP was bounded with the alpha protein. That will be changed to GTP. The GTP will come to the scenery and the GTP will leave. So upon binding of a hormone or a ligand, so the structure of the GPCR protein will be changed in a certain way. So the structure, the conformation will be changed of that alpha part. It will no longer be available for the GTP. The GTP has a specific kind of structure, but due to the change of the structure of the alpha subunit, it will no longer be available for the GTP to access the part. Now the conformation is changed in certain way, so the GTP can come and bind to the alpha subunit. So when this thing will happen, the GTP will be released, the release of the GTP, and it is exchanged with the GTP. So now this is the active state, and here that protein, that alpha GTP, will be disassociated from the complex, from the trimeric complex. So beta and gamma will be here, and it will be disassociated from the trimeric structure, from the heterotrimeric structure. Now there is another story. It will go and activate the adenylyl cyclase, which will form the AMP, which will transform the AMP to the CAMP. That will go on, and the downstream will happen. The signaling, the GPCR signaling will happen in such way. And here is another protein that is called the RGS. So if it is in a GTP bound form, if the alpha subunit is in a GTP bound form, then the signaling will be turned on. So it can't turn on for so long. It can't be turned on so long. So there should be a regulation. It can't be turned on for all the time. So it should go back to its inactive state. That should happen. So here the RGS is the enzyme that is responsible for the hydrolysis, for the GTP hydrolysis. This RGS will come and hydrolyze the GTP that is present on the alpha subunit. So at first when it was activated, then there was a release of GTP. The exchange, that was the release of GTP and exchange with GTP. And now here it is the hydrolysis that is going via the RGS. So activity of the alpha subunit is terminated by the hydrolysis of the bound GTP, which is stimulated by the RGS protein. And now the inactivated alpha GTP will again come and associate with the trimeric protein, that is with the beta and gamma. So now it will be in the inactive form, and again the chain will go on via induction of a stimuli. So here the which is the turn on, so binding to GTP. So the exchange of GTP or the binding to GTP and the hydrolysis of bound GTP. The turn off is happening via the hydrolysis, not via the exchange. So the option 3 is correct. So here is a statement based question. So one is the assertion and the one is reason, the conventional one. So the assertion is saying the mitogen activated protein kinase means MAPK are activated by upstream MAPKK. So this is the basic structure of the ERK or MAP kinase pathway, that is the signaling pathway. So here that is when it is in the dormant state, means the resting phase. So the MAP3K means the MAP kinase. So like this signaling you can take it as a like the stream, like the here the downstream, this one is the downstream, this one is the downstream, this one is the downstream. So that is you can say it as a cascade, as a signaling cascade. So that is the in the resting phase all are in the dephosphorylated form, that is in the inactivated form. So when the stimuli will come, that means a ligand will come and bind to the receptor. So the conformation will change in certain way. So it will phosphorylate the next one. So that is the MAP3 kinase. Means it is the kinase for MAP kinase and kinase. So kinase is related to the activity to transfer the phosphate group, and the kinase phosphatase to remove the phosphate group. That activity is to remove the phosphate group. So here after binding on the resting phase, those enzymes will not be in a phosphorylated form. So these enzymes will not be in a phosphorylated form. So this will be in a resting phase, and the signaling will no longer be passed on. The signaling cascade will not be there. But upon binding with the ligand, so the conformation will be changed in certain way. So this will be phosphorylated via the change in the that receptor. And now the MAP3 kinase is phosphorylated, which will pass on the phosphorylation to the downstream, the further downstream thing. That is the MAP2 kinase, means MAP kinase. The MAP3 kinase phosphorylate the MAP2 kinase, which this will pass on to the MAP kinase and further to the substrate. So that could be the transcription factor, and via this it will regulate the differentiation, proliferation, apoptosis, this kind of procedures. So the MAPK are activated by the upstream MAP kinase. That is true. The MAP here, the MAP kinase. This is the MAP kinase. It will phosphorylate the MAP kinase, and that will make it in a active form. And the reason is saying the activated MAP kinase are inactivated by MAP kinase phosphatase. So activated MAP kinase. So when this MAP kinase is activated, means it is in a phosphorylated form. So now the MAP kinase phosphatase. If you are using the MAP kinase phosphatase,
this will come here and will remove the phosphate group, and that will make the MAP kinase again in a inactive form. Like if there is a high expression of the ligand and there is a very high amount of MAP kinase, that phosphorylated MAP kinase means it will be in a active form. In that case, the phosphatase can come, and if it is, there should be some regulation, right? If it is not in a conventional way, if like some uh, some problems is happening there, like in the cancerous case, or some other kind of that if the signaling is not going in a good manner, some other kind of problems are happening. So in that case, there should be a regulation. It can't go on and on. So the regulation is via the MAP K phosphatase. So that activated MAP kinase are inactivated by mitogen activated protein kinase phosphatase. That is true, but it is not the reason for the assertion A. So both A and R are true, but R is not the correct thing. Is asking the activity of the colchicine. Which stage of the mitosis is blocked by the colchicine? So far this is the last question for our today's session. And before going in depth, we should know what is the colchicine. So colchicine is a alkaloid type of secondary metabolite. So it is produced from the plant Colchicum autumnale. And so far here in the picture, you can see the cell is going via the metaphase phase of mitosis. So here, if it is the mitosis, then you can say this is the metaphase plate where the chromosomes are aligned. So it is near to the center of the total cell. So here you can see the MTOC, that is the microtubule organizing center. So from this, on both the pole, you will be getting an MTOC center. From this center, the microtubule will be polymerized. So microtubule will be formed from here, and it will go and bind to the kinetical region of the chromosome. That is the centromere of the chromosome, which is the most condensed part, of the chromosome, and they are having that region is associated with a lot of kind of proteins. So kinetochore will be bounded with that microtubules, and from that association of the microtubule will happen in that way. It will go and bind to the kinetochore, and thereafter it will pull. So thereafter it will pull the kinetochore on the opposite way. So in such way, so this part of the chromatid will go here and this part will go here. So thus the sister chromatid will be dissociated and the two cells will be formed, and so the cell division will happen in that way. But so here we have seen that microtubule has a very much important role to dissociate the chromatids. But if you are using a colchicine, so the polymerization of the microtubule itself will be blocked by the colchicine. So this microtubule is made up of tubulin protein. So tubulin proteins are present in a heterodimeric form. So alpha and beta tubulin will come and bind. So each unit will make a heterodimeric form. One will be the alpha tubulin and another will be the beta tubulin, and it will be polymerized. It will make a chain. That polymerized chain is known as the microtubules. But if we are using the colchicine, that colchicine will come and bind with the tubulin in such a way so that it will not be able to get polymerized. So the chain formation, the tubulin, so they will be form a chain that will be polymerized and form a microtubules. That formation will not be happening. It's not going to be happening. So here in the pole, so you will get that MTOC in both the poles. So from there, the microtubules will not be formed. So what will happen? So the cell will rest at the metaphase stage. So the which stage of the mitosis is blocked by colchicine. So metaphase will be happening till the metaphase. The prophase will happen, the metaphase will happen, but the separation, the cell will stop at the metaphase plate. The separation of the sister chromatids will not be happening. So prophase will happen, metaphase will happen, but the anaphase will not be happening. So the option is the third one. So here, where you can use the colchicine. In practical case, the colchicine is used to stop the polymerization of the microtubules. So if you are stopping the polymerization of microtubule, just think in a such a way you want to see the chromosome, the genetic material, the DNA of a cell, and this chromosome in a metaphase stage, it will be in the most condensed way. So if you want to have a clear vision, you will get this from a metaphase chromosome. The metaphase gene is the most condensed one, and this will give you the more that great resolution of the structure and conformation of the DNA of that respective cell. So if you are having a cell culture, and you want to see the chromosome, you can stop that cell. If you are having a cell culture, then generally it will go via the cell cycle. It will multiplicate it. It will increase the number. And if we were stopping the growth in the metaphase plate, in the metaphase phase of the mitosis by using the colchicine, so it will no longer be that chromosome will no longer be dissociated, and you will have a very clear vision of the chromosome. So for the staining in the chromosome, if you want to see the chromosome in the staining case, that uh, that cell culture should be treated with the colchicine to have a very clear vision of the metaphase. So practically you can use the colchicine in that kind of cases. So far we have covered 5 questions today. Hope these questions were not difficult for you, and now you got a clear vision on at least for these 5 questions that will genuinely help you for having a good marks in your upcoming exam. So if you are feeling any kind of difficulties, you can feel free to write in the comment box. So goodbye, and hope we'll meet in the next video.